from Microbe TV, this is TWiP, This Week in Parasitism, episode 171, recorded on May 21st, 2019. I'm Vincent Racaniello, and joining me today here in New York City, Dickson Despommier.
Hello there, Vincent.
And Daniel Griffin.
Hello, everybody.
Hello there,
in
Daniel.
vivo. We are all in vivo.
We are in vivo.
In vivo veritas.
Indeed.
And we have a lovely day here in New York City.
It's gorgeous out there. I mean, it's really classic.
It's not too chilly?
Nope.
All right. And here inside we have parasitism.
Indeed.
So, Daniel, what do we have from 170?
All right. So, parasites inside me, right? That's our...
Right? That's parasites inside me.
What is that?
Well,
is that your shtick?
Monsters inside me. That was the one. Yeah.
Let me acquaint everyone who is clicking back in, remind them. And then for those of you clicking in for the first time, tuning in for the first time, let me present this case. We discussed last time a 3-year-old girl who was living in a remote area in the Bocas del Toro archipelago. This is in Panama, up near the Costa Rican border. She lives in a small village. Some of the homes have raised off the ground on these stilts. Some of them are over water with the overwater toilets and such. Some of the more to-do people in town might have tin roofs, while a lot of the homes are just thatched roofs. There's a lot of animals in the area. And this young gal came in, brought in by her mother, because the mother had noticed that one of the eyes was red and that the daughter, the young girl, was rubbing it a lot. And then the mother seemed to notice that there may be some visual loss. This girl may be having some issues seeing out of that eye. And she comes to one of the floating doctors, mobile clinics, and an ophthalmologist is there and does a fundoscopic exam. So they actually look in through the pupil at the back of the eye and describes band keratopathy. We said that's calcium on the cornea, and outer retinal punctate lesions. And we left our listeners to figure out what this might be and what might we do.
A little cryptic.
Well, you got two things going on here, it sounds like to me, but that's...
It really does? What are the two things?
One's on the cornea and the other one's on the retina.
Oh, I see, in that sense. All right, Daniel, why don't you take that first one? Certainly.
Case guesses. This is written in by Peter. Oh, he's got that word that I'm going to pronounce wrong. Ashard TWiP. I have not yet sent it an answer for a good few episodes on the long and arduous journey to the land of parasitological elation. Some of my emails seem to have veered off the cyber highway and not gotten to you. I also have been quite busy, but still do not miss an episode. I often do solve the cases and mean to write in an answer, but the next episode release beats me. I even got the recent Ascaris case confirmed by my mentor, Professor Celia Holland, tireless champion of Ascaris research. We got a link there. Part of the reason I've been busy is I've been lucky enough to be asked to be involved in the practical teaching of some parasitology to medical students at the Royal College of Surgeons in Ireland.
Oh, nice.
It is great that they see the value of including someone from an ecology background. I really enjoy it, find it enriching. Great to talk to the infectious disease consultants and laboratory technicians there. Also, as I often wonder how Ireland, more associated with emigration, compares with Daniel's New York, famed beacon of immigration. I, of course, recommend parasitic diseases to all the students and print out the life cycle pages to talk through the stages of the life cycles under the slides. Listening to the new case, it brought to mind the poem I have already sent in, Oncocerca by W.C. Campbell. I don't need your goddamn eye, I thought. Eye damage by a parasite, punctate lesions, and keratopathy all sounded good. Panama did not. So I changed my mind, and now I'm going with ocular toxoplasmosis, which I think may be more in keeping with the band and certainly with Panama. Great to hear all the female parasite heroes in recent episodes. I have not been keeping a list, but just in case these have not been covered yet, the Parasitology Journal has highlighted the work of Professor Ann Bishop, Gwen Reese, Letitia Obang, and Gertrude B. Elion recently. Finally, I enjoyed the discussion on anthropozoonoses. I myself do not like this term. The World Health Organization used to define zoonoses as diseases naturally transmitted from vertebrate animals to humans and vice versa. They seem to have dropped the vice versa, which I think may be due to the increased popularity of terms such as anthropozoonoses, which I believe originally is meant to mean diseases transmissible from living animals to humans, although many reverse this. And it gives us a link here and tells us to go to check this out. My problem with these terms is they give the impression that disease is unidirectional, often to the detriment of the wildlife host, as well as people not being aware that there's an infectious risk to endangered wildlife. There's also the stigmatization of wildlife. I think this is more likely to occur in an anthropocentric world where academics are under pressure to make their results sound as impactful as possible. So although I know I am in the minority, I prefer the old WHO zoonoses, diseases naturally transmitted from vertebrate animals to humans and vice versa. Salon, and this is from Peter Stewart, Trinity College, Dublin.
I kind of agree with Peter. I like this definition, the zoonosis, vertebrate animals to humans and vice versa. Why not?
Yeah.
Why do we have to have a special term?
Well, we have one for the era that we have created for ourselves, the Anthropocene. So I guess maybe there's this anthropocentric view that, I don't know.
The Anthropocene, yeah. That's the era where humans destroy the planet, right?
Exactly.
And it's the last era, right?
It's the end of Game of Thrones.
Did we do some of these heroines, by the way? Ann Bishop,
we did,
I thought we did.
Ann Bishop, yes.
Do you have a heroine for today?
If I had a book in front of me, I certainly would choose one.
I'll get you one while you're reading. I have a pile over here. You can't have it because they're autographed. They go to the listeners.
Understood.
Except if he grabs a copy, one of the new 7th edition, or pulls it down, there are more female historical parasitology figures in that.
You don't have any printed versions of those yet, right?
You know, I actually have one printed.
I have one too, but unfortunately it's in my house.
Which would be across the river.
It is across the river.
And through the woods.
That's right. To Grandma's house. Usually.
All right. But I was just looking, Daniel, the sticker, you have to change your PWB sticker.
It's it's our old logo. We don't have an
Are you going to get the new stickers?
You got it. Yeah, we are.
Certainly.
Very good. Dixon, can you take the next one, please?
Ben writes, Dear Leishmaniacs, Daniel wasn't kidding when he said that this case was a little more difficult. I had a few initial ideas, but after digging around the literature of ocular parasitoses, I couldn't believe how many parasites had ocular manifestations. A few of the common ocular parasites immediately came to mind, like Onchocerca, Gnathostoma, and Acanthamoeba, but these either didn't seem to fit the clinical picture or weren't geographically suitable. Ophthalmomyiasis, ophthalmic cysticercosis, and the apparently very rare cases of ocular giardiasis seemed to fit the symptoms to some extent, although, as I understand, these ocular manifestations of giardia occur along with the typical GI symptoms, and these were not mentioned. I also ruled out microsporidia because Mother Nature would not allow a case that would eventually be featured on Parasitology podcast to be caused by a fungus. This left me with my final diagnosis of Toxoplasma gondii. The symptoms seemed to fit with ocular toxoplasmosis, except for the inflammation, although apparently secondary causes of inflammation are common. As per Parasitic Diseases 7th edition, PCR would be the best way to confirm the diagnosis, but I'm uncertain whether this PCR testing would be possible in rural Panama. Treatment would be with pyrimethamine and sulfadiazine, which is common for toxoplasmosis, but I believe corticosteroids are also common in ocular cases. I'm not very certain of this diagnosis because the list of parasites that could cause these kinds of symptoms is so long. The weather here in the only city where hamburgers are both food and its inhabitants, it's sunny and 14 degrees C. Keep up the fantastic work.
Dickson, you know what he means by hamburgers?
I do, of course.
Where do you think he is? What country?
He's probably in Germany.
Oh, 2 recent malaria paper suggestions.
So in Hamburg, Germany, to greet one on the street of another hamburger, they would say hummel hummel, and the other person would say moss moss.
Are you serious?
Dead serious.
All right.
What does that mean? What is that? In case I'm ever there.
No idea.
Okay.
We have 2 potential papers here. Authors use correlative storm SEM microscopy to investigate the knob structures that allow P. falciparum to adhere to endothelial structures.
Wow.
And the authors show the cyclic AMP signaling controls part of the secretion of the apical organelles during erythrocyte invasion.
I'll be darned.
You like malaria, Dickson?
I love it. I love it. It's a complicated subject, but I love it.
Our next, oh, by the way, there's a paper that Ben sent in. I haven't read. It's about a possible first virus of a malarian parasite.
Wow.
I'm trying to decide whether to do it here or on TWiP.
A double-stranded RNA virus?
I don't know. I didn't look at it.
Okay. Because all the other protozoan viral infections are double-stranded RNAs.
I understand.
Mm-hmm.
I would, if I'm going to vote, I don't know if I'm allowed to vote.
What are you going to vote on?
I was
what the diagnosis is?
No, I was going to say I would love to do that on this show.
No, we can do it here. It's fine.
Yeah.
There's no reason not to.
Exactly.
All right, next is from Kevin. My father had a perplexing expression he would use to describe suboptimal choices or outcomes. Quote, better than a sharp stick in the eye.
I remember that. I remember that.
Heard that.
I don't have enough water. It's better than a sharp stick in the eye.
That's right.
Yeah.
As a child, I found this phrase unsettling. For some reason, TWiP 170 exhumed the memory. I've got to say that this case really TWiP'd me up. The minimal clinical details offered, coupled with the arcane and overlapping terminology in the ophtho literature made for choppy PubMed surfing. This whole enterprise was a bit of a poke in the eye. Our patient has a chronic eye malady best characterized as a uveitis. I believe that the other observations of band keratopathy and retinal lesions are due to the primary uveal inflammation. The word uvea, collective term for the iris, choroid, and ciliary body, is Greek for grape. The uvea is a frequent site of chronic inflammation, which can extend beyond the uvea to involve the cornea, lens, vitreous, retina, and optic nerve. Uveitis is divided into anterior, intermediate, the vitreous, posterior, and panuveitis. The terms choroiditis, chorioretinitis are often synonymous with posterior uveitis. I think that our patient has an anterior uveitis. Our 3-year-old girl is not described as blind, but her problem could be the beginning of progressive visual deterioration or the development of amblyopia, lazy eye. Corneal blindness is 20 times more common in low-resource nations than in wealthier nations. Globally, the most frequent cause of corneal blindness is trachoma. Other common causes of corneal disease and blindness are bacteria, fungi, locally prepared toxic eye remedies, leprosy, measles, xerothalamia, vitamin A deficiency, ophthalmia, neonatorium, chlamydia or gonorrhea infection, herpes simplex infection, and toxin exposure, such as, for example, Lyme, that is calcium chloride.
Hydroxide.
Thank you, hydroxide. In previous battle zones, such as Cambodia, landmine ocular trauma is a frequent cause of childhood corneal trauma and subsequent blindness or visual deterioration. Bringing the discussion back to the parasite realm, it is unlikely that our patient has toxoplasmosis, as this is usually a posterior eye disease. She does not have the common keratitis secondary to visceral leishmaniasis, case reports, nor does she have salt and pepper retinal lesions, rarely reported in Giardia infections. Onchocerciasis is not reported from Panama, and the neighboring region has very low, if any, transmission. I doubt that she has cysticercosis, though this can cause uveitis. The syndrome DUSN, diffuse unilateral subacute neuroretinitis, doesn't fit here. DUSN is a posterior disease believed to be due to a wandering nematode in the subretinal space. That's an interesting phrase. A wandering nematode in the subretinal.
I like to visualize that. They wander.
Yes, of course, it doesn't have anything to do with our little thing today, but it's nice. I will make the reluctant guess that our patient has acanthamoeba infection of the cornea with uveitis and anterior chorioretinitis. Disease acquisition is via exposure to contaminated water, ocular trauma, and contact lens use in higher resource non-poverty settings. Diagnosis is most accurately achieved by PCR. Culture yields inconsistent results and requires prolonged incubation. Smears have poor sensitivity. Confocal microscopy has shown good sensitivities in diagnosis. In poor resource countries, all of these modalities are limited in availability. Treatment decisions are difficult due to the lack of controlled studies. Topical miconazole, variconazole, propamide, and neosporin have been used. The opacification due to calcium deposition, band keratopathy, if it is affecting vision, can be treated with EDTA. Keratoplasty may be required in late-stage infections. Prevention requires the ability of clean water for washing, drinking, etc. Thank you for your valuable and important podcast, which is helping to keep the airwaves at least partly filled with knowledge and reasonableness. The 7th edition of Parasitic Diseases is quite fine. Great new cover graphics featuring a TWiP worm. Thanks for all the education. And then, of course, we have references, endnotes, and a terminal curiosity. We have references all about keratitis, band keratopathy, onchocerciasis, many, many other references which you will enjoy. But I would like to get to the terminal curiosities. Many, many references here. It's lovely.
Wow.
He's prolific and encyclopedic and highly informative.
The Boston Globe article. Here we go. Stop saying that. A 2009 expression to forget by Jan Friedman, January 3rd, 2010. Now, it's true that some images of hurting oneself are time-honored phrases. Cutting off your nose to spite your face has been around since the 16th century. It's better than a sharp stick in the eye has been faint praise, at least since the 1870s. Children have sworn, cross my heart, hope to die for more than a century. And for many of them, the next line is, stick a needle in my eye. Eventually, such familiar phrases lose some of their shock value. All right.
Yeah.
We had 3 guesses, and I think all of these individuals have previously won books. Let's see. I know Kevin has. I know Ben has. Peter Stewart might not have, in which case we'll give it to him. Peter, if you won, let us know. If not, I'll send you a book.
2 people stepped back and Peter was left.
Because the other 2 have won. And Peter, if you didn't get one and you'd like us to send you one, I need your address and phone number. And others are waiting for their books. I'm sorry, I've been a bit busy, but I will get to it.
Well, you have had good reason.
There's never a good reason. I'll get to it. And by the way, where are all the other people out there? Where are your case guesses?
You know, I wonder if it was just that this was a tough case.
This was tough.
So
you're saying it takes a certain person to admit that they're stumped
and to
send it in.
Yeah, I think that people don't like to be wrong. They hate to. I find that when you ask a question of a crowd, the person who doesn't know it is usually not the person who shouts out what they're thinking. With exception.
Yeah.
It's usually, if you actually feel like you know the answer, a little more motivation to
I understand.
Get involved. But actually, I always feel like you learn more from the person with the wrong answer. And I think, wasn't it Lee Iacocca? So we learn more from our mistakes than things that we do right. And when I hire people, I hope that they're not here for a learning experience. Something along those lines.
Right.
Dickson, do you mind being wrong?
Never.
Because you often are, right?
I think so. Yes, I am.
I am too. I'm wrong every minute.
To err is human. Remember that. To forgive, nearly impossible. That's what I used to say to my kids.
Well, I like the expectation of gratitude is
miserable.
Miserable.
Yeah. That's a good one also.
Really is. My guess
tough.
on this, I think this is acanthamoeba, by the way.
I would agree with Vincent, but the supposition that acanthamoeba only causes one thing was the red herring in this one, because I think that what you would expect to find in a let's say a contact lens user would be this little halo of erosion where the amoebae actually came to rest against the side of the contact lens on the cornea. And that's what you see. You see these little circles of erosion. But in an area where there couldn't possibly be any contact lenses because there's no advanced medical services in that area, then you have to come up with another way that this presents itself. And this must be more than common if this is the case, because it's found all over the world and it can infect lots of different kinds of people, but indeed lots more in children perhaps, because they spend a lot of time in the water playing, whereas the parents are out working, whatever. That's the assumption at least. So I would agree. I think my best guess for this one, given the paucity of information, is Acanthamoeba castellani.
So I'm going to give you guys a little more information.
Yeah, please do. Like, tell us what it was.
couple things to make you, and I'll be honest, this was something where I spoke to the ophthalmologist, and often, you can't know everything. It's like the show in ER where you know everything. You look at it and say, oh, I know what that is.
Right.
But an ophthalmologist, particularly one experienced in the tropic, will look at something and say, oh, I know what that is. And there were a couple of features that drew them. So I'm going to hit this. So one was there were these punctate lesions that we described. I'm going to be toggling between our TWiP 171 show notes here and this wonderful chart that someone handed us, sent in the TWiP 170 email.
It was Kevin, yeah.
Kevin. And so there was not an ulcer. There were not ulcerations. There were these retinal punctate lesions. So several of them, not as though there was one localized thing causing this. There was also calcium. So you got to think about something, and the progression here is there was some sort of infection that initially triggered a red eye, but now when we're seeing them, this has progressed to the point that there's been a calcification and that there are these retinal punctate lesions. So just sort of thinking it through before we hit the actual, what might it be? Let's discuss acanthamoeba as a possibility first. Mm-hmm. Is that going to just get better on its own? So that's going to usually progress without treatment. Right. So instead of the girl having a red eye initially and now it's better and she's coming in because there's visual loss, it's going to be a red eye and now it's progressing and getting worse.
Yep.
So the nice thing I like, I'm going to go to our chart here, is that we've got a pathogen. Let's see where we are dealing with the eye. So it's not an orbital cellulite. It's not sort of around the outside. It's not just the conjunctiva, but we actually seem like we're getting into the subconjunctival space and we're getting calcification. So thinking about which pathogens might end up in this girl that goes to those places. The Toxoplasma is something that we joked a little bit about, the nematode wandering about. That's not going to give us just this small barren keratopathy. That's going to, if it should die there, we've talked about the granuloma type lesion that we'll see. So that would be a sort of a retinochoroidal granuloma. And so, should we hazard another guess of what might affect the eye and lead to a calcified lesion?
Toxoplasma.
Exactly. And so I'm gonna, and actually this was the nice thing from the ophthalmologist with us. This had a classic appearance where he just basically said, this is what the toxoplasmosis of the eye looked like. And so that is, sort of to reiterate, that's one of the great things is, always think, oh, we need diagnostic tests, but sometimes what you need is experienced clinicians, sure. People with the training, with the familiarity, so they can recognize this. And I think sometimes we describe like the serpiginous lesion and saying, well, you don't need to do a test for
Right.
cutaneous larva migrans. You see it, you've seen it a few times, you recognize it. And I think when you were working in the parasitology lab, you always make the comment, a lot of it is you see it a lot of times and you come to recognize it. There's a pattern
You
recognition.
become familiar. This is absolutely right. But I've never thought of Toxoplasma as an invader of those 2 cell layers on the cornea ever. But but you can also quote me on this. I mean, I remember saying many times that Toxoplasma gondii is the world's greatest parasite. I know the virologist will stand up and say, oh yeah, but no, this parasite infects every single mammalian species and every cell type in every mammalian species. And this is great proof of that, actually.
Yeah, I think you say most successful, right? It's most successful.
It's most successful because, and also in most cases, it's a benign infection so that it's well tolerated by host and it doesn't debilitate for the most part. In this case, this poor little kid had vision loss.
How did the child get this infection?
Well, let's talk about that. How do you get toxoplasmosis? There's a couple different ways. It's not uncommon that people get toxoplasmosis. So, shall we go through the life cycle a little bit?
Sure. Let's go through. It's pretty straightforward, actually. It's a foodborne illness for humans for the most part. Although not entirely, because you can be exposed to the stage that comes out of the definitive host as well, which is the cat, which produces oocysts that then sporulate in the environment and become infectious. But you still have to eat them. I don't think you can catch this by rubbing it into your eye.
So do you have to eat them after they've sporulated?
Yes. If you eat them before that, but they sporulate almost immediately upon contact with air. So that's the trigger for them to sporulate. So you have to eat raw meat. Raw or undercooked meat is the way you catch toxoplasmosis.
So if you had a cat,
the
cat was infected.
Yep.
And so these sporocysts are in your house.
Yeah.
Is that the right name?
Yeah.
Sporocyst.
No, oocysts.
Oocysts.
They're oocysts.
Now, we're not going to, well, some people might pet the cat and then touch their mouth. Could they ingest it that way?
Little kids, sure, absolutely.
Could you get it from a break in your skin?
No.
It has to go into your gut tract.
That's correct.
And so rubbing it on the eye is not happening.
It's not happening.
So we're going to get emails about this, which is great. And it's interesting. I think that the cat exposure is often overplayed. And I've had patients where let's say they have a cancer, they undergo chemo or whatever might be happening, and then suddenly they're told basically to have absolutely no cat contact if there's a cat in the house, and the cat lovers are like, well, where are we going with this? And, and I think I mentioned one time about there is a certain time. So let's say the cat actually, defecates into the cat box. Some people actually have these little boxes full of stuff and the cats go to the bathroom in the house and then it gets scooped out. And I was, talking about how if you just change those on a regular basis, there really isn't a lot of transmission, among cat owners. Right. The pregnant woman, it's always the concern that you get toxoplasmosis during pregnancy and the different trimesters. There's different risks of transmission and also different levels of severity of infection to the fetus should that happen. But I really have to say with Dickson here is that most of this is oral ingestion. Most of it is undercooked meat. I think the transmission from the cats is probably a lot less common than,
it is.
And it's, and this is, you go and you clean the litter box after you've let the stuff sit there for a while till it becomes infectious. And then you don't wash your hands afterwards.
No.
I mean, right? It's,
That's true. But I do recall an outbreak among a writing academy in, I believe it was in San Francisco. The weather was bad, as is typical the case in the wintertime for San Francisco. And so this dressage group had to practice indoors. And this indoor horse track, had in the back of it, the food for the horse, which is hay. And in the hay, there were mice. And so they used to keep a lot of cats to eat the mice so that they wouldn't become a problem. But there was so much dust kicked up by the horse hooves that it aerosolized the oocysts. And they ate them. They didn't eat them. They breathed them in.
Yeah.
But they came in on the mucous membranes and they eventually got swallowed and they became infected with Toxoplasma.
Yeah, that's, when we were going through
Very unusual.
at one point, like generating slides of, okay, which different parasites can be transmitted different ways.
Yeah.
And actually, toxoplasmosis is considered something you can inhale when it is aerosolized. Like
Exactly.
that. So that can be a bit of an issue.
Quite remarkable. Probably not the case here, though.
No, probably not the case. Here, it probably was undercooked meat.
Yeah.
And you're only going to see this in an area where you have the felines as part of the life cycle. It'd be pretty hard to maintain this without that.
Where is this little archipelago again?
So this is in Panama.
Right.
And this is up near the Costa Rican border. It's on the Atlantic side.
They wouldn't possibly have any jaguars there, would they?
I don't think close to any of the Nobe villages, but
if they had it in the area, that's another animal that could carry this infection, by the way.
40 million people in the U.S. are infected.
Yeah, it's in France, almost 80%.
Did you know it's one of the 5 neglected parasitic diseases in the U.S.?
We did know this.
What are the other 4?
Well, I would say Ascaris, Trichuris, hookworm, and Strongyloides. How's that?
I think
Chagas,
Wittenhotes put, so I was going to,
Chagas, cysticercosis, toxocariasis, toxoplasmosis, and trichomoniasis.
Really?
Those are the 5.
And none of the other worm infections are neglected?
They're not in the U.S.
They're not
this
even
is just
known.
in the U.S. This is just U.S. neglected.
Okay. All right.
But they're in other countries, they're other neglected things.
Sure.
But I appreciate you bringing that up because we always think, or many people always think, I don't know if I always think, but many people always think of, these are diseases that are not a problem in a place like the United States. But you just listed 5 where, Chagas, we say 300,000 people living in the U.S. probably have that.
Amazing.
What was the number for Toxo? Did we say 500,000?
In the 40 million in the
40
US.
40 million, right? That's higher than 500,000.
It is. But remember what I said, this is a benign infection in 99% of the people. So
True.
Yeah, it's a problem in pregnancy. It's a problem in people with immunocompromise. Right. Take the population of France and multiply it by 0.8, and that's the same in Brazil. It's about 80% of the Brazilian population.
Yeah.
So these infections are considered neglected because they get little attention.
Exactly.
And very little action to surveillance, prevention, and treatment. So CDC is trying to raise awareness.
So are we.
And did you know that April 14th was International Chagas Day? We missed that here on Twitter.
Didn't know that, but we could celebrate.
You know how many people in the U.S. have Chagas?
We say over 300,000.
That's right. How about cysticercosis?
That's a little lower, but there's an endemic center in Venezuela that people immigrate from, and I think that's probably where it's coming from mostly.
But what number is the CDC giving us, Vincent?
They're not.
Yeah, no, it's a difficult one to determine.
But they say 1 in 10 people who have it
have symptoms
will die
from it.
Yes,
that has.
And it's preventable, right?
Never heard that one. That's an interesting statistic.
It's cysticercosis. Now, it's outside the United States. It's the number 1 cause of seizures.
That I didn't know.
Toxocariasis.
Yes.
70 people a year in the U.S. are blinded.
Yeah.
Most children.
Yeah.
Mm-hmm.
The Toxocara was last time, right?
Yes.
Toxocara canis or cat eye.
Uh-huh.
Toxoplasmosis is a leading cause of death from foodborne illness in the U.S. Trichomoniasis. 3.7 million people in the U.S. have trichomonas infection.
It's a lot, right?
It's a lot. Never going to be 0.
No.
This was Toxoplasma, acquired most likely by eating undercooked meat.
Yes.
And I guess that people started throwing off some potential remedies. What do you guys think? Should we be giving this child any kind of medicines at this point? Dickson, were you sort of implying that most people end up controlling this disease? Does this child need medicines?
They're sick. This child is not well. This child is suffering.
Are they sick now, or are they suffering the consequences of the immune reaction to the parasite?
The calcification, certainly, but the punctate lesions on the retina, it indicates some kind of an active process. Yeah,
actually, these are old. Like, this is old. When the child initially had the red eye, that was the active inflammatory response. So, Unfortunately, at this point, when we step in, this is permanent. The toxo has sort of reached the end of its road.
You just have to hope that none of them were in the macula.
Fortunately, in this child, it was not. So there was visual disturbance. It's going to be very hard in a 3-year-old to know what degree, but as they get older, visual testing, this could be followed.
You didn't confirm the diagnosis in any way, right?
No. And there.
Did you treat?
No treatment and nothing.
The only treatment I would think about, I mean, of course, you'd think about the trimethoprim sulfamethoxazole method, but also the corneal damage. You could do a corneal transplant and restore vision if that was the problem, which would be later on after they became.
Potentially. And I guess the other side is, let's say you had made this diagnosis at an earlier during the acute. Could, therapeutics in combination with maybe an anti-inflammatory agent such as the steroids, they might have had a role.
Right. I have a question. And that is,
certainly.
It's an exotic question, perhaps, but a child develops a disease that damages their cornea, and so this is a child, let's say 2 to 5 years old. You perform a corneal transplant.
Mm-hmm.
Does the cornea then continue to grow with the child as they grow older?
I don't know. I actually have not been very, very involved with the corneal transplants. Usually we think of them as occurring in older individuals.
Exactly.
Let me read a couple of terminal curiosities from Kevin.
Right.
He writes, in a conversation, if I mention that I have contributed a letter to This Week in Parasitology, the listener invariably thinks that I'm interested in telekinesis and spoon bending. I find this strange since I associate the word parapsychology with the 1970s and never hear it in its speech anymore.
Exactly.
If you look at the quantitative word usage curve for parasitology in the Ngram viewer, you will see that it peaked in 1976. TWiPlets, please continue your TWiP work. The public must stop mixing up parapsychology and parasitology. That's pretty funny. Addendum to my list of zoological medical metaphors in medicine, TWiP 165, leopard skin, dermatologic depigmentation finding in chronic onchocerciasis mentioned in PD7, page 556. And Annals of Obsolete Medical Terminology. I recently learned of an archaic term for blindness, gutta serena, an old term for amaurosis. Who says amaurosis anymore? Actually, I do. In my lecture, the last lecture in my virology course, I talked about Lieber's congenital amaurosis, which has been cured or may be cured with viral gene therapy. It's approved by the FDA. Lieber's congenital amaurosis. Let's see, what else here? Other terms which I will not define but have a nice ring to them. Seclusio pupillae, iris bombi, and Senechiae. Well, this is just lovely. And finally, I want to point out that Elmer Pfefferkorn died last month.
Oh.
Did you know that, Dickson?
Oh.
We talked about him here on TWiP once.
He was a wonderful gentleman.
He was 87.
Wonderful parasitologist, and he worked on Toxoplasma. That was his organism.
He did work on Toxoplasma.
He was a former virologist converted to protozoa.
Yes. And we had a letter written about him.
He was a clever man.
Wrote she had gone to Dartmouth and he taught a class, and she was in medical school. She called him the kindest man on the planet. Yes,
I would believe that too. Unless she had known William Traeger, and then they were tied for first.
Was he kind?
He was extremely kind.
Are you kind, Dickson?
I think so. I'd like to think I am.
You think he's kind, Daniel?
He's kind of what?
I think so.
He's never nasty.
He's very generous. Very generous.
I was not here on
very
Friday, and someone was here and took a picture of this room and said, I came to visit the famous Twix suit, and there was a box of tissues on the table. And someone on Twitter said, the tissues are for when Vincent is not nice to Dickson.
That's showbiz stuff, folks. Don't think he's not nice to me in real life. He really is. Very generous also.
No.
No, you are. I'm
not a nice person, no.
Vincent. Don't say that on the air, for God's sakes. But it's not true anyway.
I'm okay. But under certain circumstances, I can be tough.
Well, that's different.
All right, let's do a paper.
You bet.
Are we doing the paper or the hero? We're doing the paper first?
Yeah, we do the paper.
The Dinoflagellate paper?
We usually do, right?
I think so, and I've got it right here.
We do the paper.
I think we do the paper, and then a hero, and then a new case study, and then some emails.
Yep.
and then we go to dinner.
There you go.
Is that a plan?
That's a plan.
Now, I picked this paper, and my colleagues are not particularly thrilled with it because it involves no human disease-causing organisms.
No,
you're okay with it?
No, I think it's a fantastic paper. What I was saying is it just, as we started talking a little bit about it before the show, or something we talked before the show, just let our listeners know. It just seemed like the complexity of this whole area was just, you could just go deeper and deeper.
You could.
So I'm excited to talk about this.
So I hope that you too, if you ever did not like a paper that I picked, you would say, no, I don't want to do this. Right?
We're too polite to do
No, we've done that, right? Sometimes Dickson has picked a paper, and I've just been like, what is that all about?
You don't have to worry about hurting any feelings. It's not personal.
It's
right?
Exactly right. It's not personal.
So
except if I pick a paper, because I am sensitive.
Okay.
That's true.
We have to do
this is all true. This is all true.
This one, I picked this because I'm very interested in mitochondria, and that's what it's about. It was published in Science Advances. And it's called an aerobic eukaryotic parasite with functional mitochondria that likely lacks a mitochondrial genome. And so parasite there in the title. So TWiP, right?
Yep. Got it.
Mitochondria in the title. So
and I like mitochondria.
It's a parasite. It's got to be a parasite.
Now, the word likely in the title really bothers me. I don't know why they made them put that in there. Right?
I wonder. That really catches me.
I bet the reviewers made them do that because I don't think the authors would have put likely.
Have you ever had a reviewer make you change a title?
No, I have not.
I don't think I ever have either.
I haven't published so many papers, right?
Enough to have that happen if it would have happened.
First author is Yui John, and the last author is Gurno Glockner. He's come from all over the place. The Alfred Wegener Institute, the Leibniz Institute, these are both in Germany, Department of Genetics, University College London, Singapore Center for Environmental Life Sciences, University of Paris, what else? Another university, CNRS in France, Sorbonne, the Botany Department, University of British Columbia, and American University in Cairo, and the University of Cologne. Wow, what a collection. Okay, what's this about? So it's about a dinoflagellate and a parasite of it. And the parasite is what is the topic of the paper that has issues with mitochondria. In particular, it doesn't have a genome. Now, remember, mitochondria arose many, many years ago, billions right, of years ago, when bacteria invaded a cell and stayed there, and the bacteria became mitochondria. And most of their DNA left, and they became the energy-producing organelles of the cell, of eukaryotic cells. And we know also that in plants, other bacteria came in to produce chloroplasts, right? So this has happened at least twice.
And we talked about chloroplast first, mitochondria second.
We think that chloroplast happened first. Why do you think that, Dickson?
Well, because the oldest living organisms we can find on the planet are plants.
Well, bacteria are.
Well, they're cyanobacteria.
But plants came afterwards.
Oh, they're single-cell plants. They're algae.
Well, yeah, that's right.
Yeah. But I guess it was the colonial. We started in an anaerobic environment, so you really wouldn't have, They
used oxygen as a waste, as a way of getting rid of something, right? Hydrogen.
It was a byproduct of photosynthesis.
That's right? Exactly right. And that accumulated in the atmosphere, but it just oxidized everything first. So it took almost 3 billion years for all of the metals in the environment to be oxidized before the atmosphere began to accumulate O2. And at that point, we think, after that, the mitochondria arose.
Right, so we have chloroplasts and mitochondria, which are energy-generating organelles, which originated as bacteria.
The chloroplast doesn't generate energy.
What does it generate?
It generates ATP and glucose.
What do you think ATP is?
Well, that's energy, but
I was kind of going with that.
But no, the energy is locked up in glucose. It links carbon atoms together. That's what
makes glucose, which is used to build other things.
We make ATP by splitting it. It takes energy to link it.
We make ATP by splitting what?
Glucose. We eat the glucose and we split it into single carbon molecules, and
Chloroplasts conduct photosynthesis.
And we oxidize it and get rid of it.
Captures sunlight energy, converts it into ATP and NADPH, and freeing up oxygen. Right?
It doesn't even mention glucose.
Now, it doesn't mention glucose.
But that's how it's done.
Yeah, the Calvin cycle.
Yes.
They use the NADPH to make organic molecules from carbon dioxide in the Calvin cycle.
Right.
And you've got all this energy stored in the carbohydrates, which we
find delicious.
Yeah, we like carbohydrates.
So let's talk a little bit about the host of this parasite.
Indeed.
The host is a dinoflagellate.
A dino. Could you say dino?
Dino? Is it dinoflagellate?
I'm sorry to say.
I like Dino, though.
Well, it's Dino Martin.
I'm Italian. Dino. Alveolates are a diverse group of eukaryotes, and there are 3 phyla. Dinoflagellates? Dino or Dino? Dino. Apicomplexans and ciliates.
So apicomplexans are like
malaria and toxoplasmosis, right?
Right. And cryptosporidium.
And giardia?
No,
cryptosporidium. And now ciliates, we have some parasites that are ciliates.
One.
Giardia? No, and Balantidium coli, right.
Right.
And are any dinoflagellates parasites that we know of?
No. None.
Now, a dinoflagellate.
Not flagellates of human beings. So I'd say there are dinoflagellates that are parasitic, but not parasitic of human beings.
Oh, okay.
Because they break, there's like there's 4 groups of our dinoflagellates. There's the ones that are getting energy from, so the phototrophs, right? Getting it from photosynthesis.
Yes.
And then there's the heterotrophs, the mixotrophs, and then there are parasitic.
That's right. Yes, many are photosynthetic, some are mixotrophic, right? So these are unicellular protists.
Right.
They're often in the oceans.
They're algae.
They're marine
basically,
plankton.
They're algae.
This thing called protists.
It just means single cell.
Single cells, yeah. They can be found in salt water and fresh water.
Right.
And so that's a dinoflagellate.
In fact, they are the basis for the food chain in the ocean.
What eats them?
Filter feeders.
Like whales?
Well, like an organism that has a 30-foot wingspan. Can you name one? A 30-foot wingspan. A manta ray.
Really?
Yes, and they're filter feeders. The biggest organisms on Earth were plant or herbivores, which includes the whale shark, which is a filter feeder, right? And lots of other animals like that. But all of the mantid series are filter feeding organisms.
This is what really...
And sardines are filter feeders.
Sardines?
Yeah.
Okay.
A lot of the pelagic...
So the sardines eat the dinoflagellates.
Right.
Dinoflagellate. And who eats the sardines? Tuna?
Tuna.
And who eats the tuna? Whales? No, whales are filter feeders.
Just us and the Japanese.
Whales eat krill, right?
Yeah, that's right. Well, no, whales eat a lot of other things too. They eat herring. Even though they're baleen whales, they
eat Keith herring?
No, they don't eat Keith herring. They eat just plain herring.
So what I found amazing was that the genome of dinoflagellates are 10 to 100 times larger than the human genome.
Are they polyploid? No, really. I mean, if you look at it that way, then is it a lot of repetitive sequences?
They do have a lot of repetitive sequences, yeah.
Okay.
But it's still huge.
All right.
All right. Now, what's important here is that several species of dinoflagellates can make toxins and make harmful algal blooms.
Correct.
Now, Dickson, I know those are called HABs. You know all about HABs.
Well, something.
What causes a HAB?
What causes a HAB? Depends on what you're smoking. No, that's not true.
Besides the dinoflagellate.
Dinoflagellate. So the cycle for these algal blooms off the coast of Florida mostly, and we had a lot of them last summer, by the way, were triggered by something, and which is something that most people wouldn't even believe, let alone realize that this is what's going on. And what's going on is these Sciroccos, which are powerful windstorms across the Sahara Desert, which lifts up all the grains of sand, or lots of them, and a lot of dust associated with that. It's iron rich, and it blows all the way across the ocean. Some of it settles in South America on top of the rainforest, which actually provide nutrients for And the other part of it falls along the coast of Florida and Cuba and places like that. When the iron in the Sciroccos-laden soil falls in the ocean, it deregulates the growth of the algae because iron turns out to be a limiting factor in its nutrition. Right. And that's when you get these huge things. Now, we can relate this to something, right? Anybody who's gone to the University of Alabama knows full well what
Crimson Tide.
The Crimson Tide.
They call Alabama the Crimson Tide. Why?
They do, because they want a name when they lose. Because they lost to Clemson last year. But they call it, well, that's the red tide. That's basically what they're referring to. When the red tide comes in, and I believe it's Gymnodinium, and I don't remember the species name for it, but I used to remember it. When that red tide blooms, all the filter feeders die. Mm-hmm. And all the organisms that feed on the filter feeders die.
Yeah,
and so you had
these blooms are associated with toxin production, right?
Yeah, exactly.
They call them neurotoxins.
They're horrible.
They cause shellfish poisoning. Those are the filter feeders.
That's correct. That is exactly right.
They can also cause human disease.
They can.
Daniel, do you know of these
paralytic shellfish poisoning? There's also an issue with the red tides where people can have an anaphylactic type reaction.
That's right.
No, just even being close enough, they can breathe it in
as a sea spray.
Yeah, just even being close enough.
Yeah, so it's a big problem for tourism, believe it or not.
Why would that?
Why? Because when they happen, of course, the entire coastline is littered with dead organisms.
And people stay
the
away.
Smell, and you don't
smell, and then there's a succession of flies that come in on these carcasses, and they stay around for weeks, and it's a very, very sad situation for most of these resort areas.
Mm-hmm. All right. Now, this dinoflagellate, it's not actually the topic of this paper. The topic of the paper is a parasite that can infect these dinoflagellates and can affect these blooms, apparently.
Yes.
They can, I guess, decrease the extent of the blooms, right?
Yeah.
So when there's an algal bloom caused by a dinoflagellate, these parasites, which are of various names, they have a field day because they go, whoa, food. And they go inside them. And they digest them away, as we'll tell you in a moment.
So what's our famous saying about that, Vincent?
Smaller and smaller.
No, successful systems attract parasites.
Successful systems attract parasites. And so that's the subject of this paper. One of these parasites, it's an Amoebophora species.
Right.
All right, and these can infect many of the dinoflagellates in these blooms, and they can have an effect on the persistence of the bloom.
So I'm sorry.
Amoebophora, right?
Yes.
Is that the way to say it?
I believe so.
Okay, what were you going to? I'm sorry.
I was just going to make a connection between this seemingly esoteric subject and back to human disease. There is one organism that's not a dinoflagellate. It's an amoeba flagellate. And it's Dientamoeba fragilis. That's the parasite. And it does cause disease in humans.
It's a dinoflagellate.
It's not a dinoflagellate. It's an amoeba flagellate. And that's what this parasite turns out to be.
This Amoebophora?
Yeah, because remember, we'll get to the life cycle and then you'll explain it.
So here's the life cycle.
Right. It's pretty simple, actually.
So there's an infective free-living stage.
Right.
Called the dinospore with two flagella. Right? That attached to the host cell. It enters the cytoplasm. The flagella go away.
They go away. Which
is reminiscent of Leishmania.
Of an amastogote, so that
it's reminiscent, but it's
gone.
They're both flagellates. One is, of course, a hemoflagellate, but yeah, it's reminiscent. You're right. The biology is similar.
Dickson, the parasite that comes out of the snail and swims.
That's a Siccaria.
And then when it hits us, it loses the
tail,
but it's not a flagella. It's different, right?
That's right.
But it's kind of an analogous situation. The tail falls off, right?
It's true.
It penetrates your skin through a hair follicle.
It does.
All right. So anyway, back to the dinospore. Attaches to the host and enters the cytoplasm, loses the flagella, gets enclosed in a parasitophorous membrane. Now, what else? What other parasites do that?
Many. Well, Toxoplasma, the one we just talked about, did that.
Does malaria do it?
Yeah. It's not a parasitophorous vacuole. It's, because they only infect red cells.
Mm-hmm.
So you would still call it, well.
That's interesting because red cells don't really have, red cells lack a nucleus. They're a little bit,
Right. They have spare membrane parts sitting around that this parasite takes advantage of, so it's not quite the same.
But it is interesting. I remember we had a heated discussion at one point. I was looking away, and you had a heated discussion, you too. And but it was about the issue about the invasion is not into the cytoplasm. It's you're inside the confines of the cell.
That's right.
But not actually within still in a separate space. And and then what happens next was just kind of exciting and frightening for me, but you know.
This parasite then goes into the nucleus.
It escapes the parasitophorus vacuole.
Right. Goes in the nucleus and it starts to digest the host. It gets bigger as it does that. And then the cell wall breaks down of the host and releases all these vermiform stages, and they go on to divide into hundreds of new infective dinospores.
Now that sounds like an apicomplexan.
It does?
That part of it sounds like an apicomplexan. Yeah, it does, because that's what they do.
So the host, which is the dinoflagellate, they call this a dinospore because it infects dinoflagellates.
Yeah.
We always use that word host, right, with parasites, but almost victim in this sense. Okay?
Food.
Well, this is almost viral because the host cell is destroyed.
Yeah, no. I mean, the next time I invite someone over and I'm hosting someone, I'm going to make it really clear that I'm using that in the non-parasitic.
That's right. That's right.
Right, so
we're all going to benefit from a symbiotic relationship.
That's the life cycle of Amoebophora, and in particular Amoebophora serrati. That's the topic of this paper. So that's what it does. And what they did in this paper is to sequence the genome. And the bottom line is this parasite has lost not only its plastid, which is a membrane-bound organelle found in it's where you do chloroplast.
For any sort of energy production. So it could be a chloroplast. You could think of as a mitochondrium as a type of plastid, right? A membrane-bound. You'd say it's restricted to chloroplast?
I spent 3 years at Rockefeller in Christian Dudu's lab working with Miklos Miller, and they always call them subcellular particles.
Subcellular
particles, right. So you have
particle is pretty generic.
Peroxisomes and lysosomes and mitochondria. They threw that into the mix as well.
But it's interesting. They have neither. They don't have
neither. That's right.
They have no chloroplast. They have no mitochondria, but still they're able to accomplish.
Well,
which cells are you talking about? These have mitochondria, but no genome.
No genome in the mitochondria.
They do so they're aerobic, and they do aerobic respiration. So the mitochondria are functional, but they appear to have lost the genome. So it doesn't have a plastid either, so it doesn't do any photosynthesis.
The genome of the host cell that has these two mitochondria produce all the proteins in the mitochondria.
Yeah, the genome of the Amoebophora.
So aren't those
produces everything that's needed in the mitochondria?
Isn't that the mitochondrial genome transported to some other place?
Presumably the mitochondrial genome is all in the host nuclear genome.
It's integrated into the genome, which makes it
But this is where I felt like it was getting the deep complexity, which I hope wasn't taken as dismay. But and I don't know if a lot of people realize this, but the genetic code in the mitochondria is different than the genetic code in the nucleus.
It is,
which is it's interesting. We always say, how is it that the same the same three code for the same protein, but that's not true. There's a different language in mitochondrial ribosome versus.
And in some mitochondria of certain organisms, at least one of the stop codons is actually translated into an amino acid. So it's even more. Yes.
Yeah, it even gets more. Yeah.
And so there are viruses of mitochondria, and they have a different codon usage compared to viruses. Yes, they have a codon usage that matches the mitochondrial, because that's where they replicate.
Yeah, so it's not so easy. You can't just take the mitochondrial genetic code and just splice it into the nucleus, unless you're also going to somehow splice in ribosomes that will know to translate the amino acids that way, or change the language, translate it from mitochondrial language into...
So is that RNA editing? Would you call that RNA editing?
This is something completely... This is evolution. I mean, this is, right?
But would RNA editing allow for that, though?
It could, but it would have to be extensive, right? Because the code, we're talking about the code.
Sure.
So they sequenced the genome of the genomic DNA from dinospores. okay?
Right.
It's 90 megabases, 90 million bases, which is pretty small. It's much smaller than the human genome, obviously. And it has 20,000 genes, 39 transfer RNAs. It's interesting that it's not terribly reduced. They have a lot of genes. Because often when organisms go into other organisms, their genome gets reduced, but this one is not, and I think it's because there's a free living phase that probably needs some of these.
Yeah, no, that's something we've talked about before. It's like with bacteria, when bacteria becomes an endosymbiont, something along those lines, when they just have one phase and they don't need, there's an advantage. There's a selective advantage to having a small genome. But when you need the ability to both live inside the cell, but also a free living phase, what a lot of parasites have, you really can't give up any of your genetic material because you're going to need it to succeed in both environments.
Yes. They say they can find genes for a variety of metabolic pathways, amino acid biosynthesis, purine, pyrimidine biosynthesis, which of course are made for nucleic acids, fatty acids. And they say this demonstrates the limited dependency of A. cerati on its host. So it just needs it to make more.
The dinospores that are made inside the infected dinoflagellate before they burst out and go into the free living stage, do they now possess 2 mitochondria? Or do they generate them as they're being thrown out of the cell?
So here's how it works. So the answer is the dinospores have mitochondria, yes, and a nucleus. But when the cell breaks down of the host, it releases this vermiform stage.
Right.
And then that divides into hundreds of dinospores. So I think that the vermiform has the mitochondria, and then they divide. Wow. Right? There are only 2 per cell per dinospore.
Right.
Not a lot. All right. So here's the cool part. First of all, they look for plastid genes, and they don't find any.
Right.
Because we know what genes are on the plastids, and so they say there's probably no plastid, and they're related organisms to this one that don't have plastids either. So that's cool. But what the neat part is, these are cells, these are aerobic cells. They carry out respiration. They use oxygen. And the mitochondria work. People have looked at that before. But they don't see any mitochondrial genes. They see no mitochondrial DNA. Now, one of the ways you can look at this is by sequencing. In most cells, there are many more mitochondria than nuclei.
Right.
And so the mitochondrial genes are at higher copy number. In fact, that was how they did the first Neanderthal sequences. They sequenced mitochondrial DNA because they were present in such higher amounts in the old bones compared to all the contaminant. And they said, but we don't see any mitochondrial DNA here. And they even stained the cells with a dye, and there are mitochondria there, but they're not staining with the DNA stain. There's no.
So how do you generate a mitochondrion without its genome?
Oh, you just go buy them.
Oh, okay.
Well, you make it up with the components, right? The genome, anyway, of mitochondria, Dickson, are very, very small.
Understood.
So they only encode for the respiratory components.
They divide.
Membrane, right?
They divide, don't they?
They do divide. So all the components come from the host cell.
So that's where the membranes come from when they divide.
Oh, yeah, they come from the host cell.
So where do they come from here? There must be genes that encode the membrane proteins.
Oh, yeah, they're all in the nucleus of the cell.
So then how does this thing assemble?
The mitochondria?
Yes.
Well, they divide. By binary fission.
No, stop it. In this
there was
organism,
one mitochondria years ago
and
it's dividing ever since.
That's why there's only one Eiffel Tower. Did you know that? Because it eats its young. We can go on this.
What is your question?
My question is, when the dinospores are created by this vermiform
binary fission,
where are the mitochondria at that point?
Well, they're in the veriform.
They are.
Yeah, and then they just divide by binary fission. That's how mitochondria reproduce.
Doesn't that require a genome?
No, it doesn't require a genome. No.
Not within the mitochondria. It could be within the nucleus.
Yeah, it's in the nuclear genome of the cell. Yeah, it does require an instruction set, but that's in the nucleus.
So my question is that, okay, you've got all these proteins that go into the mitochondrion, but they're in the nucleus. And now I want to make a mitochondrion. How does that assemble?
It doesn't assemble. It divides.
You mean, isn't there a portion of this life cycle that doesn't have a mitochondrion?
No. It does. They all have mitochondria. Because you don't make them from scratch. Even us, we don't make them from scratch. Sperm and eggs have mitochondria.
Yeah, like we all get all our mitochondria from our mom.
Yeah.
We don't make any fresh, and those have to divide.
They divide, that's right.
And it divides because...
So our mitochondria are very old.
No, stop it. How do even our mitochondria divide?
By binary fission.
Yeah, but what elicits that?
Well, they grow and they split in half. It's like a cell grows.
They double their genome and then they split, right?
Yeah, but the genome is minimal. It doesn't encode the whole mitochondrion.
Not the membranes?
No.
And here's an example, right, of no genetic material at all, but the mitochondria.
So, mitochondria, right, they have this interesting membrane.
Why can't we grow them in vitro without anything else? This membrane of mitochondria are full of these respiratory centers, which are major protein assemblies, right? They look like little viruses.
And then there's ATP synthase, which so all of these components make a proton gradient, and then ATP synthase uses the gradient to make ATP. The vast majority of all those proteins, there are many, are encoded in the nuclear genome, even in us.
Got it.
And the mitochondrial genome, which in us is about 16,000 bases, encodes only a small number of those proteins. For reasons that we don't understand, the mitochondrial proteins are a mix of mitochondrial encoded proteins and nuclear genome encoded proteins. And that's why I'm interested in this paper, because here for the first time we have a mitochondria without a genome. So it shows that it can happen.
Okay, so my question is still basic then. What protein encodes the membrane of the mitochondrion?
First of all, the membrane is a lipid bilayer, right?
That still encodes protein, though.
Which is encoded by host cell enzymes. Its synthesis is made by the host cell.
So the mitochondrion is constantly growing? Is that what you're telling me? When it reaches a certain size, it splits?
It grows and it splits, yeah. And then the enzymes the proteins that are in the membrane are encoded in these organisms in the nuclear genome. Now, a more interesting question, which was raised in our pre-TWiP discussion, is are these proteins that end up in the mitochondrial membrane, are they translated inside the mitochondrion or in the cytosol of the cell?
Well, I guess basically that was my
That's
question.
An interesting question, and they don't answer that in this paper, right? All we know is that all the genes are in the nucleus that encode these complexes.
By laser surgery, if you ablated those two mitochondria, could they be regenerated?
No. Cell would die. You got to have a template. Actually, I don't know the answer to that.
Okay.
But if you ablated them, could they make new mitochondria from scratch?
Right.
No.
Or could you transfect?
No, I don't think they can. And I guess I would think of it this way: is the linkage between the mitochondria and the genomes in a situation where the mitochondria does have genome. The genome is not driving the division; it's actually coordinated and synced with. So it's not like the mitochondria are constantly swelling and then they become so big they pop in half. It's much like a cell. It goes through a cycle where it enlarges. There's a synchronization where you're making, the new genome, and then there's a segregation and a division. So it's much like, think of it as a cell, but this is occurring within a cell.
So we have a military operation going on outside. So you have these dinospores, which are biflagellated with two mitochondria, and they're looking for another dinoflagellate to infect.
Correct.
Can these divide without the dinoflagellate?
What divide?
The dinospores. Or do they absolutely need to infect a cell to divide?
Oh, they have to infect the cell because they all come from the...
So they're obligate intracellular parasites.
Yeah, they are.
Mm-hmm.
The vermiform divides into hundreds of infective dinospores. So the veriform stage can divide. I don't know what that must have mitochondria, though. So I think you cannot make mitochondria from scratch. No.
Okay.
Anyway, so they say there's no physical DNA in the mitochondria. You don't find any mitochondrial genes. And they go through some technical discussions to make sure they're not missing something. They show that these respiratory complexes, which are the protein complexes that are present in the mitochondrial membrane, they're all encoded in the nuclear genome. All of them. In fact, two of them are not even present. They're replaced with a different respiratory complex, which is structurally different, and those are encoded in the genome. right? So all of the mitochondrial genes are gone because the proteins are provided by nuclear genes. All the respiratory complexes are provided by nuclear genes. And they actually think that so complexes, 2 of the genes are actually look like they came from another organism by horizontal gene transfer. So essentially, there's no mitochondrial genome because the cell doesn't need it anymore. All the genes are in the nucleus. And I wonder if they were there first, and that's why they were lost from the mitochondria, right? Mm-hmm. And didn't go from the mitochondria to the nuclear gene. So those are very interesting questions. So parent transfer to the nucleus of all essential functional mitochondrial genes and the resulting loss of the mitochondrial genome. So they think that they all got transferred to the nucleus and then the mitochondria lost its genes. And that transfer may have been just accidental, but it allowed the mitochondria to get rid of its genome. And so that could happen in us. That's why I like this. In other organisms, you could eventually lose the mitochondrial genomes if those genes went into the nucleus. Now, some people think that the reason... So here's a little discussion about this. It's tough having a mitochondrial and a nuclear genome because the nuclear genome can be, if it makes errors during replication, it can be corrected by error pathways. The mitochondrial genome has a DNA polymerase, but it doesn't error correct as well as the nuclear genome. So there's a mutational disparity between the two, which is not good because you can't have a protein complex which is made of proteins encoded in nuclear and mitochondrial genes mutate at different rates. So that's a tricky question. And so in light of that, it's weird that there are still mitochondrial genomes. We don't really understand why that is. But I like this as an example of you don't need to have a mitochondrial genome. Now, maybe because it's a simpler organism, in quotes.
This doesn't sound too simple.
2 mitochondria.
Yeah.
1 cell.
Yeah.
Well, they're a low-energy group.
So that's the story.
To be continued, I presume.
I'm convinced that they don't have a mitochondrial genome.
No, so what's the next?
The thing is, why?
So can we remove the word likely?
I would.
Say they lack a mitochondrial genome.
I would take it up. But my question is, how many other organisms out there
I would like to know that
do not have mitochondrial genomes? And we have to emphasize these mitochondria in these dinoflagellates are fully functional.
Right.
Mm-hmm.
They have all the respiratory chain in them. They're making energy.
Right.
And they don't have a genome.
Right.
Pretty cool. Did you like that?
Yes.
I'm going to get you a book so you can find a hero.
Please, would you please? That won't be difficult.
Do you want to know what was last time? Theobald Smith.
That's right where I turned to. All right.
You know, I remember that because I'm a big fan of Kilborn, who I feel gets a little bit of short shrift.
Does.
Yeah, my daughter Eloise was doing a paper on... I'm trying to remember, I think it was plague. It was plague, and he was a major player in San Francisco.
Oh, I didn't know that.
And so, sort of interesting. You think of these people as parasitologists, but some of the players also crossed over in just infectious disease in general.
This guy Bruce was a magnificent.
Bruce is an interesting character. Apparently,
he went both ways, as it were.
You know what we have to do? We have to take one of these books and we have to tag it with little, so you know where all the heroes are, so you can find them.
So
more
the
quickly.
new edition has more?
More.
The new edition has more. Originally we toyed around with having like a whole section of just the heroes, and we ended up, realizing the book was just growing and growing. So we finally reached a point where we just said, you know what, that's probably enough, and we tried to trim it down a little bit. I think Stiles is probably right after Smith. Would you say Charles Stiles? Is that
Coming up.
Okay.
He's got a long write-up because of his important role in the post-Civil War recovery of the South. Okay, here we go. Everybody sitting down and ready for this one? Charles Wardell Stiles, Ph.D., 1867 to 1941. By 1900, the American South had not yet economically recovered from the American Civil War. John D. Rockefeller appointed a special commission to look into the matter. That group could not find a specific cause for the high rate of unemployment, but they did note that most of them appeared to be suffering from anemia. In 1909, Rockefeller reorganized the commission, now known as the Rockefeller Sanitary Commission, and appointed Stiles as its head. Stiles was keen on testing his hypothesis that the anemia might actually be due to an infection similar to the one Dubini had described for tunnel workers in northern Italy some years earlier. The Commission's findings confirmed Stiles' suspicion. A new species of hookworm, together with an iron-poor diet, were responsible for inducing that condition. Stiles named the worm Uncinaria americanus, later renamed Necator americanus, the American killer. He also founded the Rockefeller Sanitary Commission for the Eradication of Hookworm that recommended a number of basic public health measures aimed at preventing further spread of the disease. Installation of latrines, the wearing of shoes, the use of tetrachloroethylene as an effective treatment for severe infections, and public health education programs. Over the next 10 years, the incidence and prevalence of hookworm infection sharply declined. Sanitation also lowered the incidence and prevalence rates of all fecally transmitted diseases, further improving the health of all those who are now able to do a full day's work. Even Mark Twain commented on this incident, having arisen himself in the South, at least, I guess, Hannibal, Missouri is considered the South, and claimed, rightly so, that the Southern people were being discriminated against by the use of this derogatory term, Southern laziness, where in fact it turned out to be due to a worm. So he's a absolutely bright shining star in the world of parasitism and public health, because the single most important thing was to control the spread of feces. And Vincent, you worked on polio. Is that spread by feces?
Sure is.
Giardia, is that spread by feces?
Yes.
I'm going to ask you questions here. I'm going to give you the answers ahead of time. The answer is yes.
Giardia is spread by feces.
Name a bunch of organisms. You can go down the list. There's legions of bacteria, viruses, fungi, protozoans, helminths that are spread by feces from one person to another. And the simple act of burying it 6 feet deep solves that problem. How remarkable. Mm-hmm. How
6 feet, that was the key.
Yeah, it was, because otherwise, if you didn't, the larvae of hookworm at least could crawl out of that hole and become a place to go catch hookworm rather than not have
pretty amazing, right? Like 5 feet, if you get down 5.
Pretty amazing.
Enough,
it is, and in some places it was impossible because the soils were the wrong type, and that's where they learned their lesson about.
Or in a lot of parts of the South, they had to do it in concrete because the water.
That's also true.
You can't get down 6 feet without hitting water.
That is also true. So, a hero nonetheless, because he was a very staunchly religious person. And he was a very straight-laced person, as this picture depicts him sitting in his military uniform, looking through his monocular microscope. But the fact is that he stuck to his convictions. And if it wasn't for the publication of Dubini, and we've already featured him in another hero, if it wasn't for him going into the tunnel at St. Goddard's and seeing all those poor people that were suffering from severe hookworm disease, then we might never have heard about this in time to make this astounding conclusion. Mm-hmm. So, luck favors the prepared mind, right? In this case, Stiles and a minister, and I'm blocking on his name, unfortunately, and I know Peter Hotez would just, tap me underneath the seat and with his toe. You know the name of this guy. Come on, tell him. Peter Hotez is a world expert on hookworm disease and loves to talk about the history. And a lot of what you just heard, I learned from listening to Peter Hotez. So.
Get that wire away from the mic. You're tapping it. Just tapping it.
I'm tapping. Oh.
I don't know why you're doing that.
I'm not doing it. It's doing it by itself.
Just causing
Must
trouble.
be poltergeist. At any rate.
All right. Thank you, Dickson.
You're welcome.
Now.
Are we ready for a new case?
New case.
Oh, brother.
Another eye?
Go. An eye for an eye.
We're done with our eyes.
All right.
But we are again going to be seeing a child, a young boy, and we're just set the scene again. Here we are. We've gone by boat to a remote village, and we're seeing a young man, less than the age of 10, who is brought in by his mother, who is concerned about an ulcer on his leg. The ulcer is painless. It's about 2 to 3 centimeters in diameter, so about an inch in diameter. It has a hard nodular border. I'm actually going to show a photo of this to Dickson. I'll let him make a few comments if he wants. Now, originally, this boy was seen by, this is a Floating Doctors mobile clinic. It was originally seen by Floating Doctors a couple months before, when the ulcer was initially there, and now he's coming back, and this ulcer is not healing, and he's being seen again. And so I'm going to actually show a picture of this to Dickson, and we're going to describe it together.
Please, let's do it.
Okay. So this ulcer, as we said, it's 2 to 3 centimeters, so it's about an inch or so in diameter.
Right.
When we inspect it, it's non-tender, but I think you can appreciate this, Dickson. There's an area of induration or hardness, slightly different color skin, that goes out probably about the diameter of the lesion.
Yeah.
And it surrounds it all the way around. There's a little bit of nodularity to it.
Right.
The lesion itself, it looks clean, right? It doesn't look like there's pus or anything like that. It actually just looks clean. Yeah. You could see that there's no undermining. It really just goes from this hardness right to the ulcer.
Would you call that a crater-form ulcer?
Some people would call it a crater-form ulcer. I think that's fair.
All right.
And there is, there's this difference in color, but it's not a bright red. It doesn't look hot or angry or fiery, and it doesn't.
And it's painless.
And it's not healing.
That defies all of the classical description of an inflammation, doesn't it?
That's an interesting issue, right? So what are the classical signs of inflammation?
There are 4 of them. And I will turn this over to the clinician to tell you what all 4 are in Latin.
Yes. Well, we always do that, right? We always have
it's a rubor.
Things. So rubor.
Well, let's do it like the Huey, Louie, and Dewey. Okay, I'll do rubor.
Rubor.
Dolor.
Calor.
And pain.
No. Tumor is swelling.
And tumor, swelling. So the dolor is pain. Your rubor is redness. Your tumor is swelling.
Calor
is heat. And then if it affects a certain area, you may get laceo-defunction.
Laceo-defunction. I went to high school.
And I always add, I like to add the extra, the 6th cardinal sign, which is often odor. It's smelly.
Odor. This has none of those characteristics.
Which is interesting. So I'm going to leave people with this. I hope our longtime listeners find this to be a bit of a gift.
It's not a daunting task.
But I'm gonna leave you with a couple of things. So what do you think it is? In this limited resource setting, is there anything that you might bring to bear that we've introduced in the past that might help you a little with the diagnosis? What might you recommend for this child? Where do you go with all this?
So where this child lives, there are lots of bugs, right?
Bugs, yes. What do you mean by bugs?
Flying,
do you mean true bugs, or we're
talking about or
flying insects of various sorts? Right?
So lots of mosquitoes, lots of bees, lots of sandflies, lots of other.
Yes, and these would have access to the boy all day long, essentially, because he's playing outside, and when he's in the home, there are no window screens or anything like that.
This is
right?
true. Yeah, this is all true.
And otherwise, his health is good?
Otherwise, he's in very good health, and this doesn't really bother him. It's his mom is not happy that this won't heal.
You good, Dickson?
Yep.
All right, let's read a few emails. All right, first one, Shawzy writes, Aloha. I'm a survivor of rat lungworm disease. This podcast's hosts were very rude and disrespectful when they started laughing about Sam Ballard's case of eating a slug on a dare in episode 149, Stranger in a Strange Land. If you jerks are going to report on such a horrific disease, you should be grown up enough, have some intelligence, and be respectful for your listeners and rat lungworm disease victims that have died or in the process of dying. You're not helping anyone by behaving that way. Now go eat a salad and think about rat lungworm disease with every bite. Mahalo, Shawzy. Gentlemen,
okay.
What do we say?
Wow.
We deeply apologize if we have offended in any way the victims of this horrible infectious disease, and we meant nothing more than a little bit of mirth on an otherwise very serious program. So I myself am sorry if that came across like that.
However, we may do wrong things, but you shouldn't call us jerks.
No, that's equally bad manners. So at least behave the way you wish we did.
Yes.
No, but I was going to say, it is, we try to often, I think, make light, and I think we do this consciously, because what we're trying to do is this, I'll call the mission of edutainment, is we're trying to teach people about the, 4 billion people on the planet, basically half the planet or so, who are suffering from parasitic diseases. And we're trying to get the education out there. We're trying to get the knowledge out there. And our hope, my hope at least, is that the dynamic on our podcast makes it something that people come back to, that they enjoy learning. And so I do apologize if every so often we cross over and it seems like we're being insensitive, because that's definitely not my intention. My intention is to create an enjoyable and educated podcast. But the real mission is to get this knowledge out here. So people, this is not Monsters Inside Me, where we're trying to sort of gross people out with what we're trying to do is we're trying to teach people, get the awareness up so that less people suffer from rat lungworm disease. So.
Right. And I dare say that the general public, in hearing that someone actually swallowed a slug on a dare, that was the amusing part. The non-amusing part was the consequence.
Yeah. So.
All right. Erica sends us a link to the Fold Scope. No, sorry. Rachel sends us a link to the Fold Scope.
Right.
Which is a very inexpensive paper microscope. It's very cool. I don't know if we've mentioned it before, but there you go. Erica sends a guess for case 165, which has passed, but she writes, I am no doctor and I am starting nursing school in the fall, so I'm not sure I am even close, but I believe it could be one of these options: hydrocele, hernia, idiopathic scrotal edema. Have a great evening.
Right.
That was for the one with a swollen scrotum, right?
I think that was the
That's right.
filariasis, lymphatic filariasis,
Yes.
Wucheria bancrofti, the young lad.
Yep. Anthony writes, hearing the folk wisdom of evicting a worm by trickery brought back memories. I heard such a suggestion here in Jersey City probably well over 50 years ago. The suggested protocol, originally European, was based on the fact, in quotes, that worms like sweets. One was to make up a cup of coffee with lots of sugar and then stand in front of a mirror, breathing deeply through an open mouth. The worm would be enticed by the prospect of a delicious meal and abandon all caution. When the worm was seen in the mirror dropping the coffee with the free hand, one was to grab the worm and pull it out quickly. I just added quickly. One more serious note. As a child, I heard linseed oil or garlic added to a dog food's diet to rid it of worms. Does either provide any relief? Might national diets that regularly include certain ingredients or spices help in the prevention, elimination, or control of worms? Might the mystical religious practice of fasting eliminate tapeworms? Those are all interesting thoughts, but I haven't heard of any proof of any of those. Fasting will just make the tapeworm hungry, but it certainly won't get rid of it unless you ate nothing for 3 or 4 weeks, which unfortunately will lead to your own demise as well. The doctor, not the doctor, but the cup of coffee anecdote was interesting as a new twist on something that I'd heard before, of course. But
we talked about this last.
Yeah, we did. None of those things work. But
there are many cultures with very spicy food, and they have plenty of worms.
But
right?
I'll tell you that's not an unusual thing to ask, though, because there was a group of people who were interested in the epidemiology of infections, and they went around the world, and they actually determined that in some rare cases, the people that were eating from this particular region were showed almost no infection from a particular infection that should have been there. So then they started to pick apart the plant life to see if they could isolate some ingredients, but I don't know what the outcome was.
So, for example, in India, some people eat very spicy food, right?
They do.
Yet there's no paucity of worms.
There's
right?
no paucity of worms, and that's true.
Would you agree, Daniel?
I would agree. I mean, it would be great, and there's a lot of ideas that, if I just eat red pepper, or I think the case where if I just do a shot of liquor right after that raw meat, it's all going to be good. No, unfortunately,
that doesn't work.
Daniel, can you take the next one? Ben
writes, Dear host, sorry for the double email, but I can't help myself. Ran across a TWiP crossover preprint where they've potentially discovered the first virus of Plasmodium and appropriately named it after Russian Mitrovska dolls, a virus within a parasite within a host cell, if I remember correctly. That's that joke where they say, oh, he's a Russian doll. He's so full of himself.
Right.
Actually, that was, someone once said, like, oh, he's a Russian doll. And I was trying to interpret what that meant. I said, is it because he's full of himself? So I'm not sure that's the actual meaning. But anyway, I think Daniel said he worked with one of the authors, Nick Anstey, at some point.
Is that true?
That is true. Back when I was looking at malaria and nitric oxide in Tanzanian children, Nick was actually one of the people on the ground collecting the blood samples that I was then doing the genetic analysis on.
So his address here is either in Australia or Malaysia.
So he bounces around. I think he's on faculty at University of Adelaide, but he does. There's a situation where, and it makes it very interesting from research standpoint, where you have people go in to do timber, to work in the timber industry, but they're not native to that area. So they go in and there's a high incidence of malaria in the laborers that go into this malaria endemic region. And so in this Malaysia. And so he does a bit of work. I know at one point we were actually trying to put together a project to study the malaria, the nitric oxide, the whole story in that context. So
it's actually at the University of Charles Darwin.
It's a great name.
Which is in Darwin, Australia.
I've been there.
Town named after Chuck.
That's right.
Dickson.
Yes.
This virus, they call it Mitochondria RNA virus 1. So it's an RNA virus. Because you asked before, is it a double-stranded?
Yes, I did.
RNA virus. So let's see here. It looks like it's a double-stranded RNA virus. Blah, 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 blah. Boy, long introduction here. Come on, let's go. What did we find? Processing of the sequences. RNA
is this one we might want to discuss in the future? Are we thinking maybe? I would like to discuss.
Yeah, we can discuss it.
Why don't we?
So it's an RNA virus. We'll wait to tell you.
So now we have a collection of RNA viruses that infect parasites. Protozoans.
RNA viruses infect everything.
Apparently. Well, that's their job, isn't it?
Job? What do you mean by job?
Well, they don't have any free living stages like this dinosporozoite.
They don't. Well, they're free something stage. They have particles that move around, right? Those are free.
I guess.
They're floating in the air. They're coursing in your blood.
They're seeking out.
They're moving around your gastrointestinal tract.
New worlds.
So we call it no biologically active extracellular.
We should call it that.
They don't have flagella.
They don't. No, they
don't have mitochondria. These dinospores, they have a nucleus in flagella. They can metabolize.
Eukaryotes.
They move.
I wonder if they have
the
an RNA
flagella
virus.
Go like this, Dickson. Watch.
The other way. It's the other way. No, they go forward with them. They use them to propel themselves this way.
Oh, interesting.
Yeah.
This has been TWiP 171. And you can find it on any good podcast player. Please subscribe for us so we know who's listening. It's very easy to do, and it's free. If you like our work, please contribute. Microbe.tv/contribute. Isn't that clever? Please contribute. Microbe.tv/contribute. You could give $1 a month. That would really help. And it's pretty painless. It's less than a cup of coffee. If you can get a cup of coffee for less than $1, then don't subscribe to us. But I think it's very hard, don't you think? Have you been anywhere where you get a cup of coffee less than $1?
I don't think you can do that much anymore.
You get a free refill. Sometimes.
When I was in college, you could get a beer for 25 cents.
Wow.
There you go.
But it was sawdust on the floor kind of place.
When I was in college, you could take the subway for 15 cents with a token.
Tokens, yeah.
I still remember.
And gasoline was 15 cents a gallon.
Amazing.
Yep.
What else? Coffee was probably a nickel a cup, right?
Steak was like they were just giving it away. And the streets were paved with gold. And
if you want to take your hand at this episode's clinical case, please do so. TWiP@microbe.tv, or just send us a comment or a question. But don't call us jerks, because we're really not.
And I'm sensitive.
Well, I became sensitive at that point, too, but I didn't want to say anything.
All right.
Daniel Griffin's at Columbia University Irving Medical Center and ParasitesWithoutBorders.com. Thanks, Daniel.
Pleasure as always.
Thanks for coming in. Dickson Despommier at Racaniello.org and thelivingriver.org. Thank you, Dickson.
Pleasure.
Are you going to do the deadriver.org?
No. Hopefully never.
But many rivers are dying, aren't they?
They are. They're being abused.
I'm Vincent Racaniello. You can find me at virology.ws. Music on TWiP is by Ronald Jenkins. Thanks to ASM for their support. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
is parasitic.